So thank you all very much for coming. My name's Ian Cockburn. I'm the Honours Convenor here at the John Curtin School of Medical Research. And this evening we just want to point you in the right direction and tell you a little bit about our Honours programme and show you where to go next if you're interested in joining us. But first of all, I want to acknowledge and celebrate the first Australians on whose traditional lands we meet and work, and whose cultures are among the oldest continuing cultures in human history. So, one more housekeeping thing, which is in the course of the webinar, we'll have time for Q&A at the end, but feel free as you go along to put any questions into the Q&A box, which is at the bottom of your screen, and then we'll answer them at the end in the panel discussion. So the running order for today is, first of all, Graham Mann, Director of John Curtin, will introduce the John Curtin School of Medical Research and tell you a little bit about it. And then myself and a student who recently finished Honours, Rhys Knowles, will talk to you about the Honours programme more specifically. And then we have a panel, we have a committee of people that run the Honours programme here, a representative from each department, and they'll just spend a couple of minutes, 5 minutes each, just talking about their departments and the kind of projects that you can do. There won't be too much detail here, but just to point you into where to go. And then at the end, I'll just again go through a little bit about how to apply and what we're looking for, and then there'll be the Q&A. So with that, I'll hand over to Graham, who will tell you a little bit about the John Curtin.
Yes, hi, welcome everybody, and congratulations for coming to this webinar. It's really good to see you here. From our point of view as researchers and knowledge workers, if you like, this is a wonderful part of our job. It's a way of us seeing the future of health and medical research in this country, and for us to make our contribution to develop, if you like, the next generation. But it's not only about the future; it's about the present. For you to come here and work with us in our teams is a really wonderful thing. You bring your perspectives, you bring your passion, your desire to build a career for yourself, but you also become research workers in our teams, and you do the things that help drive us and our research. So we really have a mutual interest in you having a wonderful experience and in you learning as rapidly as you can how to become a researcher. We hope to see some of you in the future as well, coming back as postgraduate research students. So this is a really great occasion and very important in the life of our school. The John Curtin School, I've been here about a year now, and it's been my great pleasure to be the director here, having moved down from Sydney middle of last year. And despite what a weird year it has been, I'm very glad I'm here. This is a wonderful research institute, really. We sit within a university, and we're, if you like, a standard research faculty, but we are in fact one of the larger research institutes in the country. We've always had this focus since we were founded back in the late 1940s, and what we have is a series of departments who have broad and deep programs across immunology, cancer, infectious diseases, in the science of the genome, and the science of the brain. And I know that whatever what your particular passion and interest is in science, and in science that makes a difference in the community, you will find something here to spark your interest and develop your skill and build your future. So that's really all I wanted to say. I'm sorry I can't stay for the entire webinar, and I thank Ian and all of your presenters today for what they're about to tell you. And if you have any specific inquiries that you do wish to take up with me, my door is open. I'm very happy to speak to any of you. So thanks very much.
Okay, thank you very much, Graham. So I'll carry on and tell you a little bit about how Honours works. So at a purely bureaucratic level, Honours is 48 units of research work carried out over 2 semesters. But in reality, and the honours year starts at the beginning of February, so you would start at the beginning of February 2021, and you'd submit your thesis by the end of October. But really what it's about is that you have, in that time from February to October, 9 months to spend in the laboratory or working computational to devote your time almost exclusively to doing research. And you can do this with access to world-class facilities and among really world-class scientists. So what honours is fundamentally about is about producing a body of original research. You'll do experiments or you'll look at data in a new way to discover something novel. And we hope that you'll have the pleasure, the privilege of discovering something that no one else has ever known before. And then you'll have the opportunity to report that to the science community here at the John Curtin, and also if you're lucky through publication, or if we're able to go to meetings, to even to attend scientific meetings and do that as well. So, I mean, I don't want to oversell this. For sure, what you discover may not be the cure for cancer or the vaccine for coronavirus, but even a small contribution to knowledge, to expand the circle of knowledge a little, has great value. And that is the chance, that is the privilege, if you like, of doing an honours project. And at the end of this, you'll have to write your thesis up to present your work, critically analysing your data, and you'll be examined on the basis of that thesis, describing your work and what you've discovered. So it's true for most of us, and by us, I mean your potential supervisors here at John Curtin, that we're hoping that you'll develop a love of research and discovery. And I think we all really secretly hope you'll come and stay for postgraduate research and a PhD. But we do actually live in the real world, despite our reputation as researchers. And we know that's not what everyone wants. Many people go into honours wanting to go to med school, and some of you will find that science is just not for you. But your year will not, if you're in those categories, your year will not be wasted. So there's other parts to the honours programme as well. As I pointed out, the main event is doing discovery science, but there's other things that you learn. So at the beginning of the year, you have to write a project proposal describing what you want to do. You'll have to give a couple of seminars through the year. First of all, describing what you hope to do and what you hope to achieve, and at the end, describing what you have achieved and what you really did discover. You'll also have to do what we call a journal club. You might not be familiar with this, but this is where you take a paper from the literature and you describe to the members of your department what it says, what they discover, and did they really do critically analysing it? Did they really discover what they thought they discovered? Did they really do the right experiments? So that's a really hard exercise in critically analysing data. So what I'm saying is ultimately what you learn is, yes, how to do science, but also how to communicate, how to critically analyse data. And these are really valuable skills wherever you go in life afterwards. And you learn, and you can learn them right here doing honours, okay? You'll also have to do, and also as part of the year, you'll have to do the training associated with handling GMOs, if you have an animal-based project, handling animals, handling chemicals, and so on. So to finish up this little part, I just want to say a few things about what honours is not, okay? So honours is not like undergraduate science. Your conveners, lecturers, demonstrators, they do a fantastic job here at ANU and at other universities of teaching science and teaching what we know. But nothing that you learn in a laboratory-based practical can teach you what it's like to actually work at the bench and actually to do discovery science. Another thing honours is not, it's not, to be blunt, it's not always easy, okay? It's a full-time commitment. The hours can be irregular because not all biological processes adhere to a 9-to-5 schedule. It can be stressful, but it can be stressful in a really good way, in a way that stimulates you and inspires you to work hard. And you're surrounded by a group of peers who are generally very supportive, both in your lab and within your honours cohort. So what I'm saying is, to some extent, it is hard, but it is also the pointy end of undergraduate research, and it requires commitment. So I just want to finish by telling a story that I sometimes tell at these sorts of events, which is to say that when I was a child, I wanted to be a scientist. And now, as an adult, I am a scientist. And you might think that that makes me a happy person, but you might also think at this point that I sound like a slightly smug person as well. But what I really want to say is that there was a point in between being a kid and wanting to be a scientist and being an adult and being a scientist when I really didn't want to be a scientist. And that was in the third year of my undergraduate degree. it just didn't seem to be what I had signed up for at the beginning, sort of learning facts, drawing diagrams, taking exams, endless chemistry practicals. And then in the fourth year of my degree, and I did this in the UK, so things were structured slightly differently, so I had to do an honours year, and I'm not even sure at that point I wanted to. And then I went back to my childhood love of scientists, to the idea of discovery, and I thought, this is what it's really about, being at the bench, learning by doing, having a problem which was uniquely mine to solve. So that was, to me, that rekindled my love of science. And I think to now in your undergraduate degree, you won't really have a sense of what it is to actually do science. And what honours is just this marvellous opportunity to discover what it is to do science, and then to have the opportunity to do that. And then, I mean, it's 1 year. So if you don't like it, you can move on, you can do something else. And as I say, we hope that we will give you skills and knowledge that will be translatable. But if you do like it, then you can come back and you can do a PhD and carry on that scientific journey. So I think that's enough from me for now. I'll hand over to Rhys, who will tell you about his scientific journey through honours last year, and his experience of the honours year.
Well, thanks, Ian. You make it sound relatively exciting, my scientific journey. So for those who don't know me, which I'm assuming 99% of you, my name's Rhys, and I'm studying in the Eccles Institute of Neuroscience, specifically in the Dehorter group, and we're looking at developmental neuroscience. So I did my honours last year, starting in January and finishing up at the end of the year. And that was a very, very interesting experience, to say the least. I would possibly put last year as somehow the best and worst year of my life, which is probably something you hear a lot about honours. And it's very accurate. It's a massive emotional roller coaster, which requires a lot of energy. But the amount you get out of it truly is quite exceptional, and absolutely nothing like just doing a period of exams. What can I talk about? So John Curtin is quite amazing. It's an incredible institution. I've been studying here. So I did honours last year, but I'd done a few research projects before that. And I love the building. I love the researchers inside of it. I love all of my friends who are studying there as well. It is a truly incredible place to study, and I'm very lucky that I can continue my honours research and now into PhD. So the student community is quite great, as long as you seek out friends and you put in the effort to maintain those personal relationships. Regarding, say, an application process, which might be something that's relatively important to most of you, I didn't find it particularly daunting. A lot of people who are listening might not have a supervisor, and that is a pretty large, I don't want to use the word hurdle, but it's something that everyone has to have. You can't really do honours without a supervisor, right? Now, the thing is, a lot of people are hesitant to contact academics, and I think you'll find there is absolutely no reason to be hesitant. If you look someone up, whether they're a group leader or a professor at John Curtin or any of the other institutions at ANU, and you're interested in what they're doing and you think you could make a valuable contribution, or even if you don't think you could make a valuable contribution and you just think that you might be interested in trying it out, send them an email. Most of them, if not all of them, are really genuinely lovely people who absolutely adore their jobs. So just go for it. And what's the worst that can happen? You might think it's a no, but really the worst thing is you don't get an email back. But if that happens, just send another email, it's fine. Okay, so look, I think it's an amazing experience. And what Ian said is very, very true. It's very different to undergrad. So in undergrad, it's primarily, it's driven by wanting to get grades or wanting to do this lab that's next week. During honours, you lose all of that structure effectively, and you're just given a project, which of course you're going to be guided by a supervisor, but which you can expand out and you can take in the direction that you want. And what you might find is by the end of the project, you will know more about your project than anyone else in the world. And that is just the way it is. You will end up correcting your own supervisor when they make mistakes because you've been more caught up on the literature, and that is a really, really cool experience. And Ian also pointed out that it's not necessarily the case that you finish honours and you don't want to continue in science and that year's lost. You learn so much in science and you learn so much in your honours year, not only about, say, the academic field, but also about other things like yourself, how you deal with pressure, how to live on 2 hours sleep a night for 3 months straight, a variety of different things. And they are just critical and crucial things that you need to know going forward, whether that be into, say, medical school, like Ian said, or do a PhD overseas or into public service, whatever. You're going to get a lot out of the year, whether or not it's an amazing year where you end up with 5 publications or nothing works for the entire year. You're going to get a lot out of it. And so if I could sum up honours in a single sentence, it would be the best and worst year of my life. But there were definitely more highlights than there were negatives. So I will pass back to Ian. I think that probably sums up my 5 minutes. I'm happy to take, if anyone has any questions about my experience or any of my other, anything else regarding the honours program, or even the PhD program, if you're interested, feel free to ask. I'll be hanging around for the panel as well.
Thank you very much, Rhys. I'm mildly concerned by the 2 hours sleep a night for 3 months straight. If anyone is doing honours and is in that situation, they should probably come and tell me.
That was a bit of hyperbole. I'm sorry. You're not going to live on 2 hours sleep for 3 months straight. Not the case. Definitely not.
Only 2.
Yeah, exactly. Look, the 3 weeks before your thesis, I can't guarantee anything.
Okay, so next up we'll have each of the department coordinators will talk a little bit about, just introduce their departments and department members very briefly. So I think first up is Barry. He wants to tell us a little bit about the Department of Cancer and Therapeutics.
Yes. Hi, everyone. My name is Barry Thompson. I'm the Honours Coordinator for the Cancer Department, and I'm just about to share my screen. It's only one slide, just to give you an overview of all of the different labs in our department. Okay. Good. So our full name is the ACRF, Department of Cancer Biology and Therapeutics, and the ACRF stands for the Australian Cancer Research Foundation, which is a charity that has provided our new department with a substantial pool of funds to get us up and running. And it's allowed us to recruit a bunch of new group leaders, and I'll just go through them one by one. So our chair of department is Elizabeth Gardner, and Liz has a special interest in the connection between thrombosis, so blood clotting, and cancer. And the connection is these miraculous little things called platelets, which are responsible for blood clotting, but also have a fundamental role in the metastatic process, because they allow the cancer cells to be trapped in a location before they colonize a metastatic site. And so Liz knows a lot about platelets and has fascinating research programs around the molecular mechanisms by which platelets are regulated. Now, Leonie Quinn has an amazing lab which uses Drosophila, the fruit fly, as a very powerful genetic model system to produce models of different cancer types. And so the idea is you can study the function of an oncogene or a tumor suppressor, not necessarily in a human cancer cell, but in a whole organism, the fruit fly, using very simple genetic methods. And so Leonie's lab uses this approach to study a variety of different oncogenes in different cancer models in Drosophila. So Ross Hannon has a special interest in developing new cancer therapeutics. So these are new drugs which have never been developed before. And he is actually taking these new drugs into phase 1, phase 2 clinical trials to treat various types of cancer, including hematological malignancies. And these drugs are fascinating. They block the ability of cells to grow by interfering with a molecule which is called the RNA polymerase type 1. Keisuke Horikawa has a very special interest in B-cell lymphomas, which is a type of blood cancer. And Keisuke has been studying B-cells for decades now. He's a world expert in B-cell biology. And he is trying to understand how B-cells are activated through signal transduction occurring at the B cell receptor. So in my lab, we look at a new signal transduction pathway called the Hippo Pathway, which is very important in controlling the growth of normal tissues, but is also very strongly activated in a variety of cancers, particularly the epithelial cancers. So this is the colorectal cancer, your squamous cell carcinomas, breast cancers, all those sorts of different cancer types have a very fundamental reliance on this Hippo pathway for driving the cancer growth and spread. So Ben Kwa is not yet a group leader in the department, but he is a level C researcher who has developed a very interesting new lab, which is focused on the combination of radiotherapy with immunotherapy. So you might have heard about immunotherapy in cancer. Well, Ben's got a very interesting approach of combining that with radiotherapy to really achieve dramatically improved outcomes. So that gives you a kind of flavor of the different labs in the cancer department. And I'll hand over now to Ian, I think, who will pass you on for the next department.
So I think the next person up is Tanya. So I'll just hand straight over to her. If you want to stop sharing your screen, Barry. Then. So Tanya is from the Genome Science Department. And is muted.
Sorry, I forgot to mute myself. Can you hear me now? Okay, so can you hear me? Yes.
Yes.
So yes, so we also quite a small department at the John Curtin School, the Department of Genome Sciences, Genome Biology. And just to describe different directions of our work, I've grouped our group leaders into three kind of categories based, I guess, on the sometimes on the biology that they do, sometimes on the tools they use. So we have labs which are mainly using bioinformatic approaches to study genome. We have three labs that are focused on RNA biology. And we also have three groups that are interested in epigenetic mechanisms and chromatin structure and function. And I guess we also, you know, collaborate a lot with each other, and some people who do epigenetic work often also look at RNA biology, and people who do a lot of wet lab research also use a lot of bioinformatic tools, but just for simplicity, I've structured it this way. So people who mainly do bioinformatic work are two groups, group leaders, Jean Wen and Eduardo Eras. So if you're interested in things like single cell sequencing and advanced machine learning, then Jean would be a really good person to talk to, whether you have biological background or whether you have you know, a computational background. She'd be a really good person to do honors with. She also has a huge knowledge of various bioinformatic tools, and she's absolutely brilliant with the next generation sequencing. If you're actually interested in developing new computational tools, new pipelines, then Eduardo Eras and his group would be a really interesting group for you to join. So you actually can learn how to program and come up with the new tools. He's also using the new type of sequencing technologies called long read sequencing technologies, where large molecules can be sequenced in comparison to short sequencing that is mainly used now. And his focus is alternative splicing. So if you're interested in RNA splicing and in developing computational approaches, Eduardo is definitely your man. Then we have three groups that focus on RNA biology. I'll start with Ripai. He's a young group leader in our department who is a lot of time spends in the lab. So if you go to him, you will actually learn firsthand all the methods and techniques. And another interesting and unique thing about Rhys is that he has both approaches, wet lab and the computational biology. So if you go to his group, you will be learning both sides of what's really important right now when you want to become scientist in genome biology. So his focus is the transposable elements. These are the ancient viruses, if you want, that have invaded our genome, you know, very early in the evolution of eukaryotic cells. And these transposable elements, they need to be silenced in our cells. Otherwise, they can actually cause all sorts of problems, including cancer. And the silencing mechanism that is actually silence the transposable elements are the class of non-coding RNAs, short RNAs called piRNAs. So Rhys group, using the fly as a model organism, studying the piRNA pathway. And he, again, takes students, and he's very active with having honors students every year. The other two people are Nikolai Shirokih and Thomas Price. These are the hardcore mRNA biology labs, if you want. They're more wet lab based people, and they're both interested in mRNA translation and in protein synthesis. So Nikolai is also an early career researcher, a young group leader in our division. And he's also a lot in the lab, and you will get a lot of first-hand kind of supervision from him. And his interest is in how different types of stresses, for example, lack of oxygen or ionizing radiation, affects mRNA translation and protein synthesis, and how these mechanisms quickly react to stress, and what happens in cancer. How those mechanisms don't cope with this in cancer. And Thomas Price, he's also looking at mRNA translation, but his focus is a very hot topic in the field of the RNA biology right now, is post-transcriptional RNA modifications. So I think you probably know about DNA methylation and hydroxymethylation, but it's a new, reasonably new field, is the post-transcriptional modifications of RNA and how those modifications also affect mRNA translation. And lastly, the three groups that are more focused on epigenetic mechanisms and on chromatin structure and function. Thomas Fischer, his favorite model organism is Baker's yeast, and he's looking at mechanisms in which non-coding RNAs, how do they stabilize genome, and he's looking at things like DNA repair. And also, I know he has a new project which he offers to honors students next year, is looking at non-coding RNA secondary structure and function, and also RNA DNA hybrids, how they affect the genome stability. David Tremethick, he is interested in particularly in one type of epigenetic mechanisms called histone variants, and how they control transcription and also RNA splicing. And he's also interested in the three-dimensional architecture of the genome. So he's applying some novel methods that allow you to look at distant chromatin interactions and reconstruct three-dimensional architecture of the genome. And finally, my group is also interested in histone variants, but I'm interested in a particular model of spermatogenesis. So I'm looking at how sperm cells differentiate, because epigenetic mechanisms are absolutely crucial, especially histone variants is absolutely crucial for development of mature sperm. And I'm also interested in the fact that some of those sperm-specific epigenetic regulators become upregulated in cancer. And I'm particularly studying some of those epigenetic regulators in Hodgkin's lymphoma, and we can see that some of the functions that are truly sperm-specific can be applied by those regulators in the context of cancer. So we are all interested in getting students, so please don't feel shy. Just like Rhys said, please write to us, and we will definitely be in contact with you. Thank you. Ian.
Thank you very much, Tanya. So again, we'll carry straight on to Rna, who will introduce the Immunology and Infectious Disease Department.
Yeah, hello everybody. My name is Rna Bruestle, and I have the not so easy task to introduce this department to you. The Department of Immunology and Infectious Disease is the biggest in the John Curtin and is very diverse. And this is a group shot of us, lots of people missing. So we actually have almost 170 people in the department with 16 independent groups, all working on somehow connected, but also very different projects, and in part multiple projects. So we also have a lot of students, undergrads, as well as HDR students and postdocs. And what I thought would be interesting for you to know is, because to get an idea of what's happening in immunology, we do have a department seminar. So every week, some students or postdocs or even sometimes group leaders present their work in detail in a polished talk. But we also do a journal club that Ian was mentioning this before, where like diverse papers out of the immunology literature get discussed, and it's a really interactive kind of setting. And then we have something which is called work in progress, and I think this is really important because that's where students, HDR, like PhD students or postdocs, present very quickly, very rough data to get feedback on. So, and this is where you really see how science works. So some people present things they get stuck on, or things they are really excited about, but they don't know what it means. And so this is like the real like science at its best. I just wanted to introduce these two people. So David Chucky is our head of department and always open for any kind of inquiries and questions. And Nora Jane is our administrator and very, very helpful if you have any questions. So like there could be one contact you could go. So I just thought I just give you an idea about the group leaders. So this is a picture just of the headshots of our group leaders. And as you can see, there's a lot of them, and I don't want to go like through them one by one because I don't think you can take that information so quickly, but I thought I'd give you a bit of a grouping. So basically we have a lot of projects in the basic immunology. So understanding how the immune system works, looking at particular cell populations, mainly using like FACS analysis, but also methods done by the other departments, like molecular methods, genetic methods. And there's basically two big groups here, and not everybody's in the groups. That doesn't mean they don't do work. They do other things. But we basically have a lot of people working with B cells and antibodies, and a lot of people working with T cells, and some people working with both. Oh. Yeah, so if you are in particularly interested in one of these, sorry, I get the announcements. Then I think I recommend just looking at the website, looking at the documents which will be provided to you, and see if something strikes your chord. And then the other way to divide our department is just by what we're working with. So some of us are interested in pathogens. How do pathogens interact with our body from the pathogen side, or how does our body defend us from these pathogens? And then another group of us is interested in autoimmunity, or so what happens if the immune system gets out of control and we develop diseases such as for example, diabetes. So we have here Chris Nolan and Charmaine working on type 1 and 2 diabetes. I'm working on multiple sclerosis. We have lots of lupus work going on. So there's different diseases with like different groups focus on. So giving the time, I think the best way to do that is to think about what you would be interested, have a close look at our homepage, and then make your call, contact us. Lots of these group leaders are very, very eager to have honor students, and it doesn't hurt. I mean, as Rhys was saying, the worst is no response. And it's also really important, and I would like to stress that point, that you have, that you feel that this is going to work for you, so that you find a group you have to deal with for almost the whole year. And so the chemistry between you and the group and the supervisor is a very big point of this. So I think no matter where you go in the department or in the school, you will always have a very comparable experience, but never the same as the other honor students. So it's a very individual choice. Yeah, so we also do, for example, Dan here is doing bioinformatics mainly. Gaten works with CRISPR. So as I said, it doesn't make sense to point out everybody. So Ian will let you know in a minute how to get more detailed information. That's from me.
Thank you, Anna. And so finally is Ehsan, who will introduce the Eccles Institute of Neuroscience.
Hi, everyone. So my name is Ehsan Arabzadeh, and I'm going to give you a brief overview of the research in the Neuroscience Institute. The department is named after this guy, John Eccles, who was awarded the Nobel Prize in 1963 for the work that he actually conducted here at the John Curtin School. And his work tackled fundamental questions in cellular neuroscience regarding excitability, excitation, and inhibition of individual nerve cells. Now, to understand the brain is perhaps considered by many people as one of the greatest scientific challenges of the 21st century. And I thought to give you a sense of the challenge, it'd be nice to remind ourselves of a few numbers. So each of us sitting here and listening to each other has about 100 billion neurons in their brain. So that's, just to give you a sense, is the same as the number of stars in the Milky Way. And this means something about 1 million kilometers of wiring and 10 quadrillion, if I say that number correctly, contact points between these neurons. So it's basically a 1.3 kilogram of massively parallel supercomputer is sitting in your brain. Sorry, is sitting in your head. And this is what is responsible for our senses, actions, emotions, dreams, and basically makes us who we are. Now, the way to approach this challenging problem, there are many ways of thinking about it, but the way we have organized ourselves, or the way you can think of the neuroscience research in the neuroscience department, is in a bottom-up manner, starting from genes and molecules and ion channels that give the cells their excitability, the sort of challenges that Eccles dealt with, and then stepping up in small steps to single cells, the mechanisms of connectivity between individual neurons and their action, to small neural circuits, bigger networks that are at work in the brain, and eventually to behavior. And try to link, to generate an understanding across all of these levels of processing. And that's basically what the research in the department does. So in terms of the techniques that we use, and these are used across many labs in the department, and that's why I thought instead of focusing on individual labs, I might just tell you a little bit about the directions of research. So we have methods of investigating activity of individual neurons or populations of neurons. And these include electrical methods, various types of electrophysiology, and optical methods, including 2-photon calcium imaging and other methods that I won't go into right now. And we also have many different levels of preparation. You can think of an individual cell isolated in vitro, try to visualize its activity or record its activity, all the way to intact brains under anesthesia, even under awake behaving conditions, and try to tackle to get an understanding of how this supercomputer works. Now, I won't be able to introduce all of the research in the department, so I thought I'll point to the website of the Eccles Institute, where you can see a list of all of the groups that are involved and their research. You see a few projects for each group. But just in the spirit of what this meeting is about, I thought I'd give you a flavor of the type of research that happens. So broadly speaking, you can think of 3 major lines of research. One, which composes a big part of the department, is to understand sensory processing and sensory decisions. So this would involve understanding the senses of olfaction, touch, and vision in different groups, or understanding synaptic transmission in one of these modalities, understanding cellular operations in one of these modalities, or understanding the decision-making in one of these modalities. We also have groups that are dealing not necessarily directly with sensory processing, but work with neuronal development, like the group that Rhys comes from, and he introduced, and also people who have perhaps a flavor of thinking about disease conditions. In particular, there is a focus on the retina and its diseases, the genes that are involved, the diagnostics, and the repairs. So on that note, I might just stop and point you back to the website where you can find research of all of the members of the department, not just the ones that I mentioned. Thank you.
Thank you very much, Ehsan. You can just stop there for a moment. So what I'll do next is I'll just tell you a little bit about where to find more information and what sort of the next steps are. And then we'll open the floor to questions. So I just share my screen for a moment. If you Google JCSMR Honours, then hopefully you'll find yourself landing on this page here. So this is our little honours page. Great that you've all found our honours information evening. So that's obviously the first step. So thank you all for coming. But then the main thing that you have to do is to find that research project. And obviously everyone has very briefly introduced the departments, and that's an awful lot of information to take in. But then here you can find the links directly to all the research groups in each department. So you can go into these and you can look far more closely than we can possibly tell you in the short time available what kind of projects are available. There's a couple of other little links on here. There's one here which says student research projects. And so that's a PDF book, but again, it tells you pretty much the information, albeit in a book form that you can find on the internet, on the web page as well. The only thing I will say is that these titles here are very much starting points. You know, people's projects, people's interests move on. You know, the main thing is the general research topic in the lab of interest to me? And don't get bogged down in details, and then contact that person. And as Rhys says, the worst they can do is not reply. Fine, move on, contact the next person. Don't take it personally. Some people are busy, but most people will be delighted to hear from you. Down here, we do offer a few scholarships to support you during the honours year. Not many, there are 4. We generally have an intake of around 30 students. So not everyone would get a scholarship, but then on the other hand, some people coming in would have scholarships anyway. So, you know, you should go ahead and apply for these. And again, if you click on the page, there's a brief overview. They all support slightly different things because they're bequests by individuals. So you have to check, for example, you know, that project is appropriate. So medical science will apply to everyone. And again, you can just click on that button and that'll take you to the application form. But the scholarships are separate from the main application. So here, this will take you through to the College of Health and Medicine website, which tells you about the honours application process. Okay, so we can just have a little look at this. What do I need to progress to the 4th year? Okay, you need an average mark of 70% in courses totaling 36 units, so generally 6 courses in your 2nd and 3rd year, which are relevant to the discipline. Okay, so we have a sort of minimum standard for who can enter the honours programme. Somewhat off the record, though I realise this is being recorded, the definition of what is a relevant course is defined by us, okay, which means we have a little bit of leeway around this, which we generally use to give students the benefit of the doubt if they're borderline cases, okay. But the strongest thing, if you're concerned that you might not, you might not clearly meet the criteria, the strongest thing you can do is to get strong supervisory support. So if you identify a supervisor and that supervisor comes to me and says, this person is really fired up about doing research, I hope they can be in the program, that counts for a lot, okay? When should I start looking for a supervisor? Well, as we say, this is the main thing that you have to do. Everything else is kind of bureaucratic. So you should be looking for supervisors. Now, also bear in mind, right, first in, best dressed. If a supervisor, if you go to a supervisor and they say they'll take you, then if someone comes in later, even if they're a better student, generally that supervisor will have to turn around and say, well, I've already taken, X, so I can't take you, even if they want to. So get in early, okay? And how to find a supervisor, well, that's what this is all about. Look on the website, reach out to people. Don't be afraid if their photo makes them look a bit grumpy. Most people will be delighted to hear from you, okay? So, and then you need to submit your application. And again, this very useful page, you can, depending on which degree you're doing, enables you to submit your application there. But the one thing I will point out is that you will have to supply this supplementary form, which is basically where you say that you have identified a supervisor and identified a project title. Now, no one is going to hold you at the end of the year to that project title and say that's different from what you did in the end, but you need to have discussed the project and it needs to be credible, okay? One last thing, which is that many projects in the John Curtin involve work with animals. And so if you're doing that kind of project, you have to be sure that you're comfortable with that. And not everyone is comfortable with that, and that's perfectly okay. But if you're going to do a project, it's best to know before, up front, that you're comfortable with that. So for example, my lab, we do lots of experiments with animals, and every honours student in my lab has had to do animal experiments. I did have a student come to me and say that they weren't sure whether they were comfortable with this, and what we did was we said, well, come and watch us do an animal experiment, and see how you feel about it. So, and then, is this for you, is this not for you? We rather know up front rather than at the end, rather than after the fact, that you want to change your project. So I think that covers everything in terms of applications. If you have any queries, then by all means reach out to myself. And then the other person that you can reach out to is, they're actually on this call, is Mary Claire. She has this functional email address, JCSMR GradProg, that will reach her. So that's definitely the best place to go for practical questions about application and so on. And certainly any academic questions and any query, really just direct to me. And if I can't answer it, then I'll happily direct you in the right place. So I will stop sharing. And I think now we can open up the floor, or open up to questions. So I don't know, we already have 3 questions in the Q&A, so please go ahead and add any more that you want. So first of all, the first question we had was, well, this is a hard one, which is, as an engineering student, I have to do my honours based in engineering rules, including having an examiner there, that my supervisor can be in any area. Just wondering how this ties in. And I think the short answer is that if it's an engineering thing, yes, you can have a supervisor from John Curtin, providing their department head is okay with that, but really the rules will be set by engineering. And so so you won't really be adhering to John Curtin or to medical research examinations and so on. The next question, are there any seminars and classes Oh, are there any seminars and classes we would have to take during the honours year? Again, unless anyone else wants to jump in and answer that, I think I can answer that. So you don't have a formal course. There isn't formal coursework as part of the honours year that's assessed. But at the beginning of the year, you do have to do courses around biological safety and chemical safety, and that's just a legal requirement to get you in the lab. Likewise, if you're handling animals, to do animal work, you'll have to do animal training in order to be put on someone's animal ethics protocol. But these are more training courses rather than sort of assessed courses. But you are expected to take part in, if you like, the life of the department that you're in. So Arna was talking about how in the immunology department we have department seminars, we have department journal clubs. So even if it's not the honours students giving the journal club, there are other students giving the journal club, and we have work in progress meetings. So, and then your lab will have its lab meetings, and then the school has a seminar program, and you are required to attend those aspects of the life of the John Curtin School. I'm just looking at, sorry, are researchers open to undergraduate students for summer internships or offering projects for students interested in research but are not doing honours? So you can do a biological, you can do as part of biology undergrad, you can certainly do a course, which I can't remember what it's called, but in which you can do a short project in a lab, and that's often a good, well, it's a little late now, but sometimes over the summer before honours you can do a project. One thing just to be clear about is that anything you do in that project cannot be used for your honours subsequently, because that would be double dipping. So you can definitely go to a lab, you can definitely do a project, and I can't remember what the course is, what the course code is offhand. And you can, well, it's a little hard doing summer internships in a lab at the moment, just because, partly because of coronaviruses, it's a little hard to have people in the lab in a sort of, it's very hard to do slightly more informal things like that. So that is a little tricky. But you can approach a supervisor and see whether they can make that work.
Just to add on to that, the course code is BIOL 3208 and 3209 for the 12-unit course.
So if there's time, it's a little late, but some people have done those as part of their undergraduate. Casual and part-time jobs. Yes, well, we do understand that, that you need money. Particularly to do, and you're so, in general, honours is kind of a full-time job commitment. So, say 35 hours a week. It doesn't have to be 9 to 5. In fact, it almost certainly won't be 9 to 5. And we appreciate that that you'll want to work, that you will want to work, and your supervisor should allow you to do so. But you just have to make sure that any part-time work you do is not incompatible with the hours you have to work in the lab. So that's something you should work one-to-one with your supervisor. But as a rule, I do tell supervisors that they should allow that. So yes, we certainly don't want to stop you from doing part-time work. Is it possible to be co-supervised with RSB? Yes, I think that is perfectly possible. You can definitely have a co-supervisor in RSB. RSB, of course, run their own honours program. So if you're spending most of your time in the RSB lab, you probably should be doing RSB honours, and it's not a zero-sum game. We're not trying to poach any students from there. So, yeah.
Can I maybe jump in? Just thinking of this engineer question. That's of course an option for you, no? So you could see if there's a supervisor in engineering who has collaborations with John Curtin and wants to co-supervise you that way. There's definitely some projects across engineering and the John Curtin.
Okay, if we've successfully found a supervisor, can we start ASAP in December during the summer break? So that's again kind of the same question about the internship. The answer is, I mean, you can, in the sense that providing you could do that either as you could do that as an internship, though as I say, the rules around that may be a little complex at the moment. You could do that as kind of the biology project course. But as I say, you can't apply that work to your honours degree. You can't put that work into your thesis because that gives you an unfair advantage over other students who would be starting at the very beginning of February or at the end of January. I'm finishing yes. So we do have a mid-year intake, and I think our honours webpage even has the date for the mid-year application deadline. So yes, and it's not normally a problem. In fact, no, it's not a problem at all. The mid-year intake is normally a little smaller. So normally we have around 30 people starting in what I always call the main cohort, which is a little rude to the mid-year students, and then around 10 people in the mid-year cohort. Actually, this year, because of coronavirus, we had many students from the usual cohort, the semester one cohort, if you like, starting mid-year. So we actually have equal size mid-year and semester one cohorts. And so that's yes, that works fine. I don't know if anyone wants to add anything about taking mid-year students. I mean, it may be a touch harder to find a supervisor mid-year. I graduate in 2021, can I apply now? Well, not really, because you need to have completed your undergraduate in order to qualify for honours. So if you graduate in June 2021, then you can apply for mid-year honours, or you can just wait and you can apply to start in 2022. But the requirement for entry into honours is that you have completed your 3-year undergraduate degree.
Ian, can I add to this as well?
Sure.
It doesn't mean that you can't look for a supervisor already. So it doesn't hurt to look now, even for the mid-year next year. So to line it up. Most supervisors are happy to commit that far in advance.
So here we go. What if I finish my honours degree after semester one of 2021? Hang on, do I have to wait to 2022? So you could, if you finish in the semester one of 2021, you can start in mid-year 2021. Yes, you don't have to wait until 2022. So you can. I mean, just as many of you will be finishing your undergraduate degree at the end of this year, you can still obviously start at the beginning of next year, January 2022. Notice that the deadline on the supplementary form is November the 30th, and the deadline to submit the honours application is December the 15th. I mean, I think this is just one of these things where we would rather you had everything sorted out sooner. And if you're still looking for a supervisor on November the 30th, then you've probably left it a little late. So yes, we want it in early so that we can just check, for example, that the department has room, because we always ask the department heads to make sure that they're happy for honours students to come in. So again, the sooner you get it in the rare case there might be a problem, the better, because then we can sort it out. We might have to say, oh, well, actually that supervisor, one problem which we have had a little bit at the moment is that because of physical distancing in the labs, it can be a little hard to actually fit in physically as many people as we would want. So if you get that information in sooner and there's a problem, we at least have a little bit of time to turn around and sort that out. So I mean, I would say please do get that supplementary form in by November the 30th. What contributes to our honours grades? Okay, so as I said, there's little wiggle room here. What we do is we identify the courses which are cognate to your honours discipline, right? So if you want to go into immunology, we'll look at all the courses related to, immunology, infection biology, biology in general, and they're 2nd and 3rd year courses, and we'll identify the ones which we consider to be relevant, okay? and then we take the top 6 marks you have in that area, and we take the average of them, and that average, oh, sorry, maybe I'll finish this and then I'll answer what I now realize your question is. And then we average that, and then if it's above 70%, then you're eligible. But now I realize on rereading your question that you might be asking, how is the honours grade worked out? Yes, that's a good question too, which is that the honours grade has 3 components. So the overwhelming majority, 75%, is the mark of your thesis and your oral exam. So that's the main event. In the first semester, you'll have to do a journal club, and that is graded. It's graded on your understanding of the paper, the presentation you give. That's 15% of the mark. And then at the very end, in mid-November, for the semester 1 cohort, you would give you'll give a seminar, and that's 10% of your mark. So, can you lock in a supervisor prior to the application closing date? Well, yes, you should have to. So basically, you should have agreed a supervisor. That's what I think that's what we're trying to say, is find that supervisor as soon as possible, lock in that supervisor ASAP. After the applications, we then assess all the applications, and formally it's my decision, but it's a decision made by the committee as a whole, and we look through those applications. And as I say, if there's an issue, then we can come back to you if, for example, the head of department doesn't want that supervisor to have a student or something. But as I say, those sorts of things are incredibly rare. So yes, lock in the supervisor as soon as possible, and then of course that supplementary form has to come in, and that says that you've found that supervisor. If I nominate another school to be the main honours supervisor, am I still eligible for scholarships at the John Curtin? Yes, there you go. Anna is shaking her head. No, I think it needs, the John Curtin scholarships are really for doing research here. That is the idea. And as I say, they do each have specific rules which you can look at. But yes. Considering the small intake, is the 70% cutoff competitive enough for most of the labs in JCSMR? I mean, anyone want to answer that one? No, I mean, I think normally, obviously you have to be smart, you have to meet that 70% cutoff, but normally I think there's a fairly good match between the number of applicants and the number of places. We try to be inclusive. We try and make sure that everyone who is enthused about science, meets the cutoff, can do science.
Maybe to add to this, having the best marks not necessarily means you're the best honors student. So sometimes I have made a decision to give somebody with lower marks the place, even if I had like a couple of applicants, just because I thought there's more enthusiasm behind and the right questions basically. So there's a lot more than just marks to doing honors.
Yeah, and speaking for myself, many of your supervisors, are not sort of savant geniuses. I think most of us, like many of us, have fairly ordinary undergraduate degree marks. I think what distinguishes us is again this love of doing bench science and that sort of thing. So I think we're all alert to the fact that it's not really, well, you have to be smart, not be stupid, but it is about enthusiasm and about really loving the work. Can each supervisor take more one honours student? They can. We would probably look at it fairly closely just to make sure that person is not overstretched. Conversely, an honours student can have a couple of supervisors, so you can be co-supervised across different departments. All these arrangements are possible. If the supervisor hasn't supervised someone before, we actually generally require them to have a more senior co-supervisor. Again, it's not that it's generally a problem, it's just to make sure that everyone is properly looked after. Is it possible to do honours part-time over 2 years? So the official answer to that question is yes. You are allowed to apply to do honours over at 12 units per semester, but we really do not encourage it. And the reason why we don't encourage it is because we feel it's unfair, because it's very hard for us to police essentially what what part-time constitutes and so on. So someone can say they're doing part-time and then carry on over 2 years. I mean, more broadly, I mean, it's not really, I think, what it's about. I mean, lab science, it is sort of a full-time job just by the very nature of having to look after cells, having to look after animals and these sorts of things. It very rarely is compatible with a sort of a part-time structure. So I mean, yeah, so we would scrutinize such an application very closely, I think is the answer.
But of course there are projects which makes it possible. So while it's not optimal, again, don't be discouraged, because I can see that you have little kids and can't commit full time or something like this. We will look at that. No, we're not saying no.
Yeah. What is the transition from a research project to a PhD? What is the likelihood of getting offered and accepted to a PhD program? Again, maybe Anna would like to answer that, because Anna is also our HDR coordinator. In practical terms, many people, I think, well, maybe Rhys can answer. Maybe I'll hand it to Rhys first of all.
Okay. So the way it works is, as you come towards the end of your honours, the first thing, similar to applying to your actual honours year, you need to find a supervisor. Now, most of the time, I would say, you would continue with your honours supervisor for PhD, and usually they're more than happy to do that. Other factors like funding permitting. And so you effectively, towards the end of your honours year, if you want to continue to PhD, you have to put in a physical application that's through the ANU's portal, and you have to write a research proposal. And that research proposal outlines effectively where you want to go in your PhD, and that's examined by the department, just to make sure that you have an idea of what you're doing and you know the background of the project. I will say though, if you're doing this towards the end of your honours, you usually are already thinking about your thesis. And so the research proposal is usually just the future directions component of your thesis if you're staying in the same group. So you can talk about all the different directions you want to go and what you think the results might be and why that's relevant within the field of science. Getting accepted into the PhD program, I believe there is a hard cutoff at first class honours, or at least that's the cutoff to get the scholarship, and I think that's the governmental cutoff. You can get accepted, is that correct, Anne?
Yeah, so there are a couple of cutoffs. So H1, so there is a way to even get into a PhD program if you're not H1 in certain circumstances. So your final mark is calculated relatively complex, like previous research experience, papers, your undergrad marks, your honors marks, all of this. So there's a lot to it. So it's not a clear cutoff, but scholarships are harder to get. So in H1 does not mean automatically you get a scholarship. And that doesn't prohibit your PhD or your supervisor to take you on anyways. So this is a different decision. But often if you're not, if you don't reach that cutoff for scholarship, they might not be able to finance you. So this is really, I think honors is a very, very good way into a PhD, absolutely, because if you are a good member of the team and you do good research, then the supervisor will try his best or her best to keep you on. And then there's different scholarships available, and Mary Claire could comment on this, but it's really, there's differences between domestic and international students. There's also different specific scholarships, so that's a whole different ballgame. But yes, doing an honors is a very good first step to get into that.
I mean, I think the short answer, if you say what is the likelihood of getting offered and accepted into a PhD program, if you get an honors mark above 83 or 84, which many people, I think possibly around the majority of people do, then you have a very, very strong likelihood of getting in. I mean, I think the balance of probabilities, yes, even if you get a little lower than that, you still have a likelihood of getting offered accepted into a PhD program. But yeah, so again, we aim for inclusivity. We're not trying to keep people out. What months is mid-year honours to run? I should know this off the top of my head, and I actually had to think about it and write it down. It generally runs mid-July, and then I think you submit your thesis at the end of May, and the examinations are all done by June. So how do I go about finding a supervisor? Email them, and they should set up a meeting, generally these days on Zoom, unfortunately. And that's when you can ask if they want you. I mean, I think most supervisors will probably talk to a couple of people, so they probably won't give you an answer on the spot, but they should give you an answer quickly. So yes, email them and talk to them. Look through the website, look through the book, find a topic that interests you. I'll graduate in June next year. I want to know if there's some new projects next year and when I should apply. So as I say, I mean, we actually used to have a page on the website which was called projects, and but the problem is this page was never kept up to date because projects change all the time. So again, I wouldn't think in terms too much of individual projects. Even in that booklet that we have, it'll say someone wants to work on fucosylation and something or other, right? I wouldn't be too hung up on that sort of thing. If the topic area of the lab interests you, go and talk to that person. Speaking for myself, I mean, the projects I list in that book are just very broad, open-ended topic areas. And what I'm interested in doing today is not what I'll be interested in doing 6 months. So again, the advice is always the same. Identify the general area that interests you, but then go talk to the supervisor. Don't get too hung up about specific things listed on websites and that sort of thing. How do you not take on too much for the scope of the honours project and become over-ambitious? Anyone else want to answer that one?
I can give it a go if you'd like.
Go ahead, yes.
That's a very good question. And one thing you'll probably realize when you do your honours is your research proposal is always going to be very ambitious. And then what you end up putting in your thesis will be like a quarter of your research proposal. I think it's not as simple as to say, oh, becoming overambitious and then not reaching that goal. That's not exactly how I would say honours works. At the end of the honours period, it doesn't exactly matter how big your body of research is or how amazing that data is or field changing it is, just that you have the ability to articulate it and understand it, and you show that in your thesis and in your defence. I personally think it's always a great idea to have a relatively ambitious project, because that means you're not going to finish it in 3 months. And it also means that you have a bunch of directions to go if things don't work, which is definitely important within an honours year. I think being ambitious isn't a bad thing. And just because your project is ambitious and you don't meet that, your entire research proposal or what you're planning to do isn't a bad thing in itself either. Would you like to add anything to that, Ian?
I think I was just going to say generally about the scope of honours projects. So people have to write research proposals at the beginning. They say they have 3 aims. Generally, if you can do one of those aims really well, you're fine, right? What you need is depth. You need to have done. It is better to have done a few experiments, repeated them, got really good reproducible data, than a whole bunch of stuff where nothing is reproduced and everything is messy. But your supervisor will guide you in this. And we also have a system where you have an advisor who can also flag problems if a project is overambitious. So you know, the supervisors here are very experienced in knowing what is the scope of a good honours project. So you know, you'll work out how much to take on with them. That's not normally. I mean, my entire time as Honours Convenor and before that on the Honours Committee, no one has failed to produce a thesis because they were too ambitious or because they were under ambitious. Everyone has ended up with a body of work that they can turn into a thesis. So you know, you're looked after in that regard. I'm currently a second-year undergraduate student at ANU. I'm interested in biology research project. Do JCSMR take students for research projects? Yes, they definitely do. So as I say, things may be a little tricky with coronavirus. People aren't in the building so much, but in principle, we can yes, there's no obstacle, there's no bureaucratic reason why a JCSMR supervisor can't do the biology research project. Do we mark to a bell curve? No, we do not. We have a very tightly defined set of criteria which we use for marking theses and which we use for all assessments, and we mark to the criteria. That said, we do send our marks to the college, and the college may ask to adjust our marks if they think we're being too generous. And we are, yes. So they may move the marks around. Now, again, in the entire time I've been convener, they have never adjusted any of the marks. We have given them, and all our marks are done against a rubric, which you have. You get that at the beginning of the year. You see the rubric. And actually, that rubric is really useful because it helps you to know how to write your thesis and so on. Are the scholarships academically competitive, or do they consider other factors? That's a hard one. Do you know the answer to that, Anna, off the top of your head? The scholarships have different rules.
Very tightly project dependent, so they have a certain topic.
So many of them are topics in the cancer department, which is because people left the money and they said they wanted the money for cancer research. We had no control over that. In general, it is done by a ranking of GPAs, but they're given out, actually, technically, they're even given by different people. So some of them are administered by the departments and some are administered by the scholarship committee. So have a look at the rules. But I think as a rule of thumb, it's mainly academics. Are there different considerations for master's students compared to honours students, or are they looked at in the same way? So there are various master's courses where the second year of that master's course is essentially the honours year. No, they are all basically put in the same, they're marked exactly identically. There is basically no difference. How likely is it to achieve an H1 in the honours year? In general, around, I think it's, 80% of students get an H1. So yes, it is. Now then, whether an H1 is good enough to get you a PhD, you probably need an H1 plus to get to be certain of getting a PhD. And what is the percentage of honours students who get published papers? In my lab, well, so there's different kinds of published papers. A first author paper. So 2 of my honours students have had first author papers based on their honours work. And I think 3 of them, so another one has a co-author paper. So that's and I think I've had about 6 or 7 honours students, but then some of them who finished may still get a paper. It just might take a few years before that paper finally gets finished, because everything takes a long time. So it's not a huge percentage. Well, I don't know. What would you say, Anna?
I think to get an honours and paper during your honours thesis is not huge, but to get somehow recognized for your work is actually quite common.
And again, if you do a PhD, obviously, again, your honours work doesn't contribute to your PhD, but you may then continue your honours work, and that honours work will end up in some paper that you publish as a result of the PhD. And then again, in my lab, I had a student, and their honours project went really well, but I then actually ended up employing them as a technician for 6 months, and then they finished. They really finished the honours project and got it in, worked up for publication, and they ended up with a first author publication. So it might be more the exception than the rule, but it definitely happens. So yes. Are there any courses you'd recommend taking, especially in terms of basic skills to do actual work in a lab? So again, obviously, if you want to do an immunology honours thing, it probably be as well if you had done the molecular immunology course and that sort of thing. But as I think we've also suggested, it's less about the knowledge that you have and more about your aptitude. As for actual work in the lab, again, if you've done a biology project, and you sort of know what it's like, then that's great. But then honours is really about discovering what it's like to do actual work in a lab. I mean, so yeah. And then the last question: when finding a supervisor, do you list the researcher or the group leader? Is this lab specific? It should, well, your supervisor, well, okay, so in most labs are quite small, and the group leader, the leader of that group will be your honours supervisor. That's in the majority of cases. There are a few labs, say for example, the Vinuesa lab in immunology, your supervisor is quite a large lab, and within that lab there are sort of what are called group leaders, who are sort of mini lab heads, if you like, and they can supervise honours students. And in fact, technically a postdoc can be in a lab, can be your supervisor, though normally postdocs at least have a co-supervisor with them. So what's the best way of answering this question? So yeah, normally it is the lab head. And if it's the, so you should probably approach the lab head first, and that lab head, for example, if you did approach Carola, she might say, well, I won't supervise you directly, but you know, Julia in my group is looking for someone to supervise, and she might suggest you approach them, and then you would list Julia and Eliard as your supervisor. So likewise, if you were, so I would approach the lab leader, and you might find that you end up being supervised by a postdoc in that group, but approach the lab head first, I would say.
I think you have to approach the lab head first, yeah, because they would have to sign up anyways on your, so if the person you supervises you is not a lab head, they need to ask permission anyways from that lab head. No? But this is a question you should ask when you have that contact. So who would supervise you? Because that might change your mind. No? Maybe you don't want to be supervised by a big lab head, or you want to be supervised by the boss. Yeah.
I mean, this is you know, like, is it better to be in a small lab or a big lab? And the answer is, of course, it depends. Because, you know, a big lab may have some great famous scientist in charge of it, but you may not actually see that person. But then, as I say, in most of those cases, you'll be supervised by a very experienced postdoc group leader who'll be able to look after you. In a small lab, you know, you may not have the demigod as your supervisor, but you know, you might have a bit more time to actually see the supervisor, and you might be, you know, so I wouldn't say there's a hard and fast rule that big labs are bad and little labs are good, or vice versa. It just depends on the sort of the specific circumstances. Unless anyone wants to add anything to that. Does it help to get into honours or PhD if we have papers published from undergraduate projects? Well, it probably doesn't help in the sense of honours, just because you know, almost nobody has papers. So, and if you've got papers, then you probably meet the entry requirement anyway. For a PhD, then yes, there are, the way the calculation is made for your PhD is, if I remember rightly, if it hasn't changed, it's a combination of your undergraduate GPA with something called your research potential. And your research potential is your honours mark plus bonus points, and those bonus points can be for published papers or for paid work in a lab. Anything else, Anna?
That's pretty much it. Yeah.
Yes. So it definitely helps with the PhD application. How do you know if you have great chemistry with the supervisor? Well, how do you know if you have great chemistry with anyone? I mean, you have to meet them and see if you get. I mean, yeah. I mean, you're right. It's definitely about checking that you can get along with the person. So one thing you should do, perhaps we should have said this before rather than the very last thing, is, you know, by a good supervisor. Well, again, it's a little hard at the moment, but a good supervisor will encourage you to reach out and talk to members of the lab and give you the chance to talk to members of the lab. And that's a really good way of finding out what the style of the lab is. You know, in general, I wouldn't go to a lab where they insist upon calling you, you know, Dr. Bruestle. I think we're mostly on first name terms. And yeah, and as I say, try and get in touch with the students, and the students will tell you how the lab works and so on, and tell you about their projects. And yeah, I hope that answers your. Yeah, that would be my main thing. Talk to members of the lab at that point, and then you'll get a sense for what the supervisor is like. Okay, I think that's all our questions. Thank you very much for staying, and thank you all for coming. Do not hesitate to get in touch, but you know, seize the day and start reaching out as soon as possible. So I think, oh no, hang on, no more questions. Okay, so I will leave it there. I'll thank my panelists. I will also thank Caitlin very quickly, who organized the webinar and everything, technical things. And okay, we'll say farewell.